Hey guys, how's it going? Way to make it past or through the first test. Hopefully I'll have your scores in by uh, the end of the week, if not for sure by Tuesday, and we'll go over some of the tests on Tuesday as well. So now we start a new unit, and we're gonna look a little more into how the cell works. So we'll look at how the cell divides and also how cells, well, organisms specifically, pass on their genes to the next generation. So heredity and things like that. In this lecture, we're going to look at cancer, and part of how cancer works is that it disrupts the cell cycle. So we'll also touch on the cell cycle. And then the next class, we're going to talk about mitosis and meiosis, which are two types of cell division. So let's get started with cancer. So what is it and where does it come from? Really, all that cancer is are a bunch of cells that divide in an uncontrolled manner and result in a tumor. So it's really just, it's not a virus, it's not a pathogen from the outside, it's your own body, it's cells in your own body that just go out of whack and start growing until they become a tumor. And then that tumor eventually can grow in an organ and mess it up, like your liver or your lungs or your pancreas, different kinds of cancers that happen in different tissues. And there on the right, you can see that even dogs and trees and mice get cancer. So it's not just a human thing, it's a universal disease because any cell can become a cancer cell. The way it happens is because of mutations that accumulate in genes that regulate cell division. So basically, the mutations are going to affect proteins that are important in controlling cell division. So when they're messed up by mutations, cell division is just going to go crazy, and the cells will divide in an uncontrolled manner and become a tumor. And that can happen in any kind of cell, not just human cells. All right, and let's look at how they arise. So again, they arise by the accumulation of mutations. And these can be typically two kinds of mutations. Mutations that disable genes that keep cell division in check. So it controls cell division and makes sure it's not happening in an unregulated way or an uncontrolled way. And mutations that will activate genes that promote cell division, that are actually in charge of making cell division happen. So these genes can be activated, and it's like you're flooring the gas in your car, right? And so the genes that keep cell division in check are like the brakes, and the genes that promote cell division are like the gas. So these mutations will cut the brakes and press on the gas, right? So then these cells divide in an uncontrolled manner. And it all begins with one cell. So in the figure below, you see normal cell division occurring where one cell divides into two cells and so on and so forth. And this is happening in almost every cell of your body right now. It's how you went from one cell in your mother's womb to 37 trillion cells that you are now. But every now and then, a cell will divide and give rise to a potentially cancerous cell. So this is a cell that has accumulated the mutations that we've talked about. And so then that cell, once again the brakes are cut and the gas is being pressed, then it will divide in an uncontrolled way and eventually become a tumor. And all a tumor is a mass of cells. Okay, it's not necessarily cancer yet. So a tumor can be benign if it has no effect on the surrounding tissue, if it's not affecting a vital organ. And that's a non-cancerous tumor. But it can also be malignant if the tumor is invading surrounding tissue or is has a negative effect on an organ, a vital organ specifically. And that's when we're talking about cancer. And eventually, a malignant tumor can spread, can metastasize. That's the scientific name for the tumor spreading. And this happens when individual cells from the malignant tumor break off and start a new tumor elsewhere. So you'll often hear, right, that the lung cancer that someone had spread to their brain or to their liver, and that's metastasis. So in order to become a full-blown cancer, cancer cells must disrupt a few things. First one of these that we'll look at is a cell cycle. And the cell cycle is exactly what it sounds like. It is a cycle that cells go through as far as growth and division goes. So the major part of the cell cycle is called the interphase. It is the majority of it, and it consists of 3 parts. It has the first gap phase, known as G1, and this is simply where the majority of, this is kind of where the cell is hanging out. Okay, this is like you and me going about our day normally. So this is the cell growing and doing what it's supposed to be doing. And then it enters into the S phase, where the S stands for synthesis phase. And again, to synthesize something is to bring something together to make something. So here you're synthesizing DNA. So this is where your DNA in a cell goes from 1 copy to 2 copies. That happens in the S phase. And then you have the G2 phase, which is where the cell continues to grow and prepare for cell division. So here specifically, it's gearing up for division. And the next phase is mitosis. And mitosis is what we typically know as cell division. This is the actual cell division step of the cell cycle. And we're going to learn more about that on Tuesday. But here you can see that mitosis advances and you get 2 arrows. So you've basically gotten to the point where you have 2 cells, but they're not divided yet. So the last step of the cell cycle is cytokinesis, which you see there at the end. And cytokinesis is when the cells actually pinch off. The 2 cells pinch off and become 2 separate cells. And then you start back at G1, right? And the cell grows and it does its thing, and then it goes through S phase, the G2 phase, and then mitosis again. So you can see that the reason that the interphase is called interphase is because it is the phase between cell division. So it is the interphase. Cell division is the most dramatic part of this cell cycle, so the stuff between it is called the interphase. And then also, just to I love to break words down. We'll look at this a little more on Tuesday's lecture, but cytokinesis simply means cyto is the cell, kinesis is movement. So it's the movement of the cell when it's pinching off into 2 separate cells at the end of mitosis. Okay, so cancers must disrupt this cell cycle. They must actually, what the cell cycle has are security checkpoints to ensure that everything is okay. So just like in the airport, you have TSA officers that are checking not just at the security checkpoint, but at different checkpoints to make sure that things are running smoothly and that there are no dangers lurking. So the checkpoints in the cell make sure that everything is running smoothly. And if it's not, then it's going to stop the cell cycle and prevent it from going further until those errors or dangers are repaired or taken care of. So you have a checkpoint at the end of G1 before DNA replication. And basically, the checkpoints here are proteins. All of the checkpoints that we're going to talk about right now are proteins that are ensuring that things are okay. So before it goes into DNA replication, into the S phase, these proteins are making sure that the cell is ready to go into this process. Because if it's not, if, for example, there aren't growth factors that are needed to help the cell grow and get ready for cell division, if there aren't enough nutrients, if the cell isn't large enough, then it probably means that it's doing it prematurely, right? It's doing it when it shouldn't be, and that's never a good thing. It should only happen when everything is present for it to happen. So if any of these conditions aren't met, then the proteins will stop the cell cycle until these errors are repaired or the dangers are taken care of. And so if it passes that checkpoint, it duplicates the DNA in the S phase. In the G2 phase, it begins to grow some more to get ready for cell division. And there's another checkpoint at the end of G2, which again ensures that DNA replication happened correctly. And if it didn't, then again, that means that there's something wrong with that cell cycle. Things are happening too quickly, or there was an error in DNA replication that could have introduced a mutation. So again, it'll make sure that everything's okay before it proceeds. Then if everything's okay, it'll go into mitosis, and there are actually many checkpoints during mitosis, right before the cell pinches off, the cytokinesis. And these checkpoints ensure that mitosis happened correctly. If mitosis didn't happen correctly, there were errors, it could be, again, you could be introducing mutations or actually deleting genes, rearranging the chromosomes that make up the cell, just really bad errors. And again, if there are errors, and these proteins, these checkpoint proteins, are going to stop the cell cycle until it's fixed. And if any of these errors can't be fixed, the cell will actually commit suicide, which we'll talk a little bit more about. But the cell will go through programmed cell death, where it hits the self-destruct button and it kills itself so that it doesn't spread these mutations or whatever things were going on that were making it divide funky. And so the cell will kill itself. Okay, so that's the cell cycle, and we'll touch on this more when we get into mitosis on Tuesday. Another thing that cancer cells must disrupt is contact inhibition. And basically, this is kind of like a cell's personal bubble. So normal cells will stop dividing and stop growing when they come in contact with other cells. They don't like to be too crowded. And so as soon as they come into contact with a cell, their personal bubble tells them, okay, stop growing, don't divide anymore. And that's what a normal cell is like. In a cancer cell, you can see there the second flask in the figure. Those cells have no personal bubble. Their contact inhibition has been disrupted, right? And there's genes, obviously, that are involved in this process, in this personal bubble. And so when these genes are mutated and disabled, then the cells can continue to pile on, which is not a good thing, right? That's how tumors happen, is the cells continue to divide, and they don't care if they're just forming a mass, right? Because if you think about it, in your skin, in your organs, your cells have they'll only divide a specific number of times, and your organs will remain in the same shape. Your skin you don't have mounds of skin cells that are growing uncontrolled. They have contact inhibition, so they form a nice little sheet or layer of cells. Again, in cancer cells that have lost this ability, contact inhibition, cells that have lost contact inhibition will just continue to grow and pile on top of each other and eventually form a tumor. Okay, and then the other thing besides contact inhibition is anchorage dependence. And again, if you think of anchorage, that is simply being anchored to something. And cells need to be anchored to something in order to be healthy. If a cell is just floating around, they're going to hit that self-destruct button and commit suicide, right? It's morbid, but that's actually a fail-safe mechanism. It's a safety switch, because cells should not be floating around. Again, they have proteins that make sure that they are anchored. So that's anchorage dependence. And if you think of it like a Christian, right, needs to be in community, cells need to be rooted in community. And they stay anchored to other cells and to the surface they grow on. And when you lose this anchorage dependence, this is when cells will detach and metastasize and spread to other parts of your body. So here you see cells growing again in a flask. And normal cells are anchored to each other and to the surface, but cancer cells are actually floating around. They have lost their anchorage dependence. So now, if they detach, they don't kill themselves. They can still live and continue to grow and divide. And now they're free to go to a new location and start a tumor somewhere else. So there are actually many other cell processes that have to be disrupted in order for cells to become cancer cells. But these are just three of them: the cell cycle, contact inhibition, and anchorage dependence, that I want you guys to be familiar with. So now let's look at how mutations can promote cancer, can turn a normal functional cell into an invasive cancerous cell. So mutations in cancer typically affect oncogenes and tumor suppressor genes. So oncogenes are actually normal genes that promote growth and division, and they're called proto-oncogenes before they become cancer-promoting genes. They're just normal genes. You can see there to the right, the proto-oncogene codes a protein which stimulates cell division only when the conditions are right. So it's a good protein, and it helps the cell go through the cell cycle and through cell division. But when a proto-oncogene acquires a mutation, this mutation will activate the proto-oncogene and turn it into an oncogene. So you can see there's a mutation, and that oncogene will now code for a mutated protein that may overstimulate cell division by overriding checkpoint controls, those TSA officer proteins that we looked at in a few slides back. So now an oncogene can go, and they're like the gas in your car. Again, back to the gas and the brake analogy. So this is like pressing the gas and causing cell division to happen in an uncontrolled manner. An oncogene, just what that means, onco means tumor. So this is like a tumor-promoting gene. And a proto-oncogene, proto means before. So it is before the gene, before it becomes an oncogene, before it gets mutated into an oncogene. The other genes, the other gene that's important in a normal cell becoming a cancer cell is a tumor suppressor. And a tumor suppressor is a gene that suppresses growth and division. So you can see there in the figure, the tumor suppressor gene codes a protein which stops tumor formation by suppressing cell division. So this is like one of the checkpoint proteins, right? One of the TSA officers. If something's going wrong in the cell cycle, this is one of the proteins that will stop the cell cycle. These are the brakes of the system, right? The proto-oncogenes are the gas, the tumor suppressors are the brakes. And these, when you acquire a mutation in a tumor suppressor, it typically is a mutation that inactivates the tumor suppressor gene. So it can no longer suppress uncontrolled growth and division. So again, with the oncogene, now you're pressing hard on the gas, and with the tumor suppressor that's been inactivated, you have cut the brake line and you can no longer hit the brakes. And so these cells will divide in an uncontrolled manner. And so you kind of have to acquire both of these mutations. This is called the multiple hit theory. For cancer to arise, you need to acquire multiple mutations or hits, not just one or the other. And again, tumor suppressors and proto-oncogenes are just two broad categories of genes that need to be mutated in order for a cancer cell to arise. But there's many other genes. So this is the multiple hit theory. For example, a mutation in a proto-oncogene and a tumor suppressor will give rise to cancer, but not one or the other. So here you have acquired a mutation in a growth receptor. So let's go back to the picture with the normal cell. In the normal cell, you have a growth factor, which is the orange ball there, that's attached to the growth receptor. And this is just telling the cell, okay, it's time to divide, you can go for it. And you can see that also cell division is regulated at checkpoint G1. So there's a tumor suppressor protein, the green blob there on the DNA in the nucleus that is suppressing the cell cycle, right? It's actually, in this case, it's a protein that repairs damaged DNA. So it's repairing any mutations that might occur. Now, this is a cell with one mutation, and the mutation is in the receptor. So the receptor now behaves as though there were growth factors present all the time. So this growth receptor is telling the cell to divide all the time. It's just like divide, 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 divide. And so that's eventually that cell is going to divide many, many, many times. Cell division is overstimulated, and a benign tumor will form, a mass of cells. But the tumor suppressor is still there, so it's still making sure that the DNA isn't being mutated and it's not accumulating more mutations. But eventually, because the cells are dividing so much, you'll acquire another mutation, and by chance, it might hit this tumor suppressor, and now you don't have this checkpoint. Now you don't have this protein that's ensuring that there are no mutations that are happening when the cell divides. So now the cells are dividing a ton, and they're accumulating a ton of mutations, and this is where you can actually get malignant tumors, right? So these cells acquire multiple mutations that allow them to do crazy things like recruit more blood vessels and grow to enormous size and spread to other tissues. And that's when cancer develops. So you can't just acquire one mutation and become a cancer. You need multiple hits. So this is kind of the opposite of killing two birds with one stone. This is, in order to kill a bird, in order to make a cancer cell, you need two or more stones, right? All right, so let's end with this. These are risk factors for cancer. A few of these you might know, a few of these you might not know or you need reminding of. But one of them is smoke, which is why there's a big push in the US to stop smoking, to desincentivize smoking. But smoke has very harmful chemicals in it. UV rays are also very harmful. Both of these are carcinogens. They are agents that will mutate DNA. So smoke has carcinogens in it. Specifically, smoke from cigarettes has a ton of carcinogens that will mutate the DNA in the lung cells. And so again, you're mutating DNA and acquiring multiple mutations, and that's how you get lung cancer from smoking. And then with UV rays, what they're doing is they actually induce mutations in the DNA of skin cells. So if you are one of those people that likes to get on the tanning bed, just beware that you're introducing a ton of mutations that can lead to skin cancer in the future. So those are carcinogens. The next ones are risk factors. So if you eat a high fat and a low fiber diet, there is experimental evidence, scientific studies that have shown that people that, in mice specifically, and now people that eat these kind of diets are more prone to developing cancer. A lack of exercise is also a risk factor. And then finally, aging. This is unavoidable. This is the risk factor that we will all succumb to at one point. So the reason aging is a risk factor is because by the time you're 80 years old, 70 years old, whatever, your cells have divided many, 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 many times. And we've talked about how just by dividing, by the act of replicating the DNA, your cells can acquire a mutation by chance. And so after dividing hundreds, if not thousands of times over a 70-year lifetime, your cells will have acquired mutations that can turn a cell into a cancer cell. And so just by the simple fact of growing old, you will get cancer no matter what. You live long enough, you will get cancer. And so ultimately, aging gets us all. Cancer is unavoidable in that sense. But we'll talk in class if you guys want about different treatments and different therapies that are being developed to fight cancer, which are actually showing a lot of promise. And so I know cancer is a very touchy subject. It is a disease that has taken millions and millions of lives. And so know that I am not a fan of cancer. It's a terrible disease, and have utmost respect for those of us who have had family members that have whose lives have been taken by this disease. So know that, yeah, I don't take it lightly, but it's really interesting to know the biology behind cancer and how it happens. So hopefully that you learned quite a bit on this lecture, and I'll see you guys on Tuesday. I won't assign homework for the weekend as usual. So we'll pick up back up on mitosis, on cell division next week. All right, have a great weekend. Bless you guys and enjoy Jesus.